and we'll introduce our speaker today. So we are very excited to start the new kind of academic year with invited talks for A3D3 Institute. And this talk this month is from a neuroscience perspective. And our speaker is Nick Steinmetz. Nick Steinmetz is assistant professor from University of Washington and in the Department of Biological Structure. And Nick's work and research is focused on recording the activity of neurons, both on different scales. So on large scales, many neurons are being recorded, and also developing analytical tools. How do you analyze then these recordings and make sense of them? Do they contribute to behavior? These kind of questions Nick is addressing, and he will tell us much more in his talk. So we are super excited to host you, Nick.
Thanks very much. Happy to be here, and let me know if the slides or the audio isn't working well. Otherwise, I will assume it is, and I'm happy to be interrupted at any point with questions. I know that this is kind of a diverse audience from a standpoint of what fields you're coming from. Probably not everyone here is a neuroscientist, so if I'm saying things that aren't making sense, feel free to interrupt, and I'll try to communicate clearly. So today I'm going to talk about distributed coding of vision, action, and cognition in the mouse brain. And to sort of lay the big picture framework for what I want to try to address in this talk, I want to start with this image from a review paper that was written in 2010, which shows in the little boxes with the acronyms, shows different brain regions, a few dozen brain regions. So these are regions like prefrontal cortex, parts of the thalamus, medial-dorsal nucleus of the thalamus, red nucleus in the brainstem. So different parts of the brain, and then in the lines showing known anatomical connections between them. And when we look at this diagram, I think people in this audience may note that there's a kind of a contradiction going on in this diagram, because on the one hand, we have this extremely dense and convoluted web of interconnections between areas, which people who study dynamical systems will recognize gives the possibility for complex dynamics, and certainly raises the possibility that any particular signal coming into the system or any particular signal generated by the system could propagate really anywhere in the system and could interface with any different process that's going on in just a small number of connections. On the other hand, we see in this diagram that there are these words scattered around at key locations like attention up here, and planning, and perception, and pain, etc. And that in the context of such a convoluted web of connections, that really in a way seems like it could be a contradiction, because how could it be the case that a system with this kind of structure has cleanly demarcated and divided little regions that are performing completely independent processes? So the big picture question I'm going to try to ask or address in this talk is whether it is the case that when we look at the activity of this system, the mammalian brain, whether it is the case that we find that different functions appear to be cleanly separated out into distinct regions, which would imply the existence of some sort of very sophisticated routing and delineation of functional activity system within the brain, which is certainly plausible. Or instead, is it the case that the activity in the brain associated with any of these different kinds of processes that are labeled here instead appears to be more distributed around different nodes of this network, in which case we might say that activity, that distributed activity, is more consistent with the kind of wiring diagram we see in front of us. And it sort of motivates a different way of thinking about the system rather than this way of trying to put little labels on every different parts, and instead would motivate a kind of more dynamical systems-based approach, a more well, many different routes you could take to study such a system. But that's the big picture thing I want to try to address in this talk is it the case that activity associated with any of these kind of labeled processes is in fact localized or is in fact distributed across the brain? And to get at this, in the first part of the talk, I'm going to talk just a little bit about a technology that we use to query the activity of different brain regions during behavior. And this is critical because if you only got to query the activity or read out the activity in any one brain region at a time, you would not really be able to answer this question of the extent to which activity is either distributed or localized across the mammalian brain. So let's start there, and then I will get to some scientific experiments with the technology that attempt to give the answer. So what we need in terms of a recording technology to answer the question that I raised is extensive recording of neuronal activity. We need to be able to record from multiple neurons and multiple different brain regions. We need to be able to access all parts of the brain, both, it's not obvious maybe from this diagram because and let me actually put on a laser pointer, I think. Laser pointer. Okay, so maybe not obvious from this diagram, but these different brain regions are, some of them located near the surface of the brain, but some of them located deep within the brain. And so we need some technology that enables us to access brain areas anywhere in the mammalian brain. We're going to need single neuron spatial resolution. The reasons for this will become more obvious as we go, but essentially past literature has well established that the properties, the functional response properties of individual neurons, even if they're immediately adjacent to each other within a single brain region, may diverge wildly. So we're going to need to know exactly what each neuron is individually doing. And we're going to need millisecond temporal resolution because the kind of behaviors that we're going to be talking about evolve on very fast timescales, like perception and action. You can imagine very fast loops between those that require measuring activity on a very fast timescale. And so we're turning to a technology called electrophysiology. And I'm just going to super briefly give a little bit of history here. The idea is that an individual neuron, I'm sorry I don't have a scale bar here, but individual neuron is just a few microns in diameter, about 10 microns in diameter, and generates these small electrical signals. And to record them, we can put a piece of metal down in the brain right up next to the neuron. And as long as this electrode has appropriate properties in terms of its impedance and the size of it, et cetera, you can detect on the other end the electrical signals emitted by one of these neurons. And this technology, with the ability to record from individual neurons at millisecond temporal resolution, at the resolution of individual electrical events or spikes from these neurons, that originated in the 1950s. And that's great because it gives us that single cell resolution and the millisecond temporal resolution, but we need to scale it up if we want to be able to ask this question about distributed versus localized processing. And so we started in the 1950s with just a single wire, basically, that came down into the brain. And by the late 80s, we had advanced to sort of these twisted bundles of wires called a tetrode, which enables recording from a few more neurons in a localized region and also enables better signal processing. But to scale up even further, researchers turned in, well, starting in the 70s, but sort of really coming into its own as a practical device in the 90s, to silicon fabrication techniques where you could essentially make it so that each signal coming from an individual recording site took up as minimal of space as possible, so that when this device is inserted into the brain, you can damage the tissue as little as possible. And the limit here in terms of how many neurons you can record is roughly the number of recording sites, which is roughly the number of printed wires along this silicon shank, which in this sort of 90s era technology was something like 16 wires for a device that's getting to be, say, 100 microns wide, which is getting to be sort of too big and too damaging to the neurons nearby. So in order to scale it further, we just need to really make it so that these lines that we're printing on this silicon device get smaller and smaller. And here's one example of how this was done with a printing technique, a fabrication technique called electron beam lithography from this example is from Ed Boyden's lab, where they were able to print 200 recording sites on a really small shank. So this really gets you a large number of neurons across a good span of brain tissue on a single small insertion device, which is great. But then they start to hit a different limit, which is that you have now hundreds of signals coming off of this, but all those signals need to be amplified, filtered, digitized, then ultimately multiplexed so that you can stream them to a computer, record them, analyze them later. And this was the sort of setup that this probe from the Boyden lab had associated with it. This is, you can see the scale bar here is 10 centimeters. Now, all of these signals, all of these little wires, which were printed very small on the part that goes into the brain, had to be broken out and split out to a whole bunch of different amplifiers, creating a huge device that obviously is incompatible with, for example, freely moving recordings in an awake behaving small mammal like a mouse. So just to emphasize that, I guess this is the probe itself is actually located way down here, and this is a huge thing relative to the size of the probe. So to overcome that limitation, we, as part of large collaboration in 2017, introduced the Neuropixels device. And this device looks like this. So here's this 1 centimeter scale bar. So it has a shank that's inserted into the brain. And the key feature to overcome that wiring constraint is that this shank that's inserted into the brain is actually attached on the printed silicon chip to a base, what we call a base, which is the entire amplification and digitization and multiplexing system all built onto the probe itself. So in just a few square millimeters right at the base of the, right near the mouse's head or the subject's head, all of those signals, as many as we get off of the device, are coming out and getting amplified and digitized and multiplexed. And how many signals are we getting? So here's a sort of zoom in, here's a photo, and here's a schematic. Each one of these little black squares, each one of these little gray squares here is a recording site. And there are almost 1,000 recording sites along the length of this, of which you can record almost 400 simultaneously. So recording 400 different signals from the brain, which span then about 4 millimeters, because the 1,000 sites span about the 10 millimeters. Recording that 4 millimeter span densely with hundreds of sites was a new capability that had not existed in the literature before. And this is the kind of signals that you get from this, just maybe slightly for the aficionados, but maybe of interest to anyone who sort of wants to understand exactly what the data is and where it's coming from. Each one of these black lines here is the voltage trace over time recorded from one of these recording sites. So each one of these little squares generates a signal that, or picks up a signal that looks like one of these black traces. And you can see that, of course, there's some sort of like background noise level that's sort of persistent across all channels at all time points. But most notably, there are these sort of deflections in the traces. And these temporary deflections, these very brief deflections, about a millisecond in duration, these correspond to individual action potentials, individual spikes from neurons that are sitting next to the probe as it's down inside of the mouse brain. And you can sort of see the processing challenge that we need to go through in order to make use of these data, which is to both detect the timing of these different events, but also classify the events according to their shape. You can see that this one and this one and this one are all looking identical. They have both the same shape over time and the same shape over the different channels that they're on, that the event is observed on. And so we would conclude that these three events likely come from the same individual neuron, whereas this event, for instance, comes from another neuron that's nearby to that first one in space. So that's what the raw data looks like. And again, this is just about 100 channels out of the 400 that you get to record simultaneously with a single probe. And the payoff is this kind of thing. So I'm going to talk more about this experiment in a few minutes, but suffice to say that we have a mouse who has one of these probes inserted in its brain. There's actually two of these probes. The other one is kind of behind the first one. And you can see that this mouse is awake and doing some behavior, is moving his sort of forelimbs and licking some tube. And while the mouse does this behavior, we can actually see the neural activity from multiple different parts of the brain and hundreds of total neurons, all simultaneously recorded. So in this case, this frontal probe goes through secondary motor cortex, anterior cingulate cortex, prelimbic cortex. These are parts of frontal cortex. Also goes through visual cortex, parts of hippocampus, and parts of thalamus. And so this is then essentially enabling exactly that wish list that we started out with at the top of the talk. Now we can observe simultaneously the signals and their relationship to behaviors, perception and behavior, across multiple different brain regions at millisecond temporal resolution and single neuron spatial resolution. So this is super exciting. Let's see. The labs who introduced this, including the Carandini Harris lab that I was a part of as a postdoc, are not the only ones who have successfully deployed it. This technology has had huge uptake in the community. I think something like 12,000 of these probes have been shipped to hundreds of individual labs, neuroscience labs. And so it's been able to be used by individual investigators, as in this example, Will Allen, sort of a single man team recorded a whole bunch throughout the mouse brain, but also from larger scale collaborations here from the Allen Institute for Brain Science in Seattle, and this image from the International Brain Lab, which has a forthcoming study using this technology to record many different parts of the mouse brain. So this is a highly successful and reproducibly successful technique in the literature. So I'm just going to mention briefly, just for interest, that last year we had this paper on a new version of the device that we call Neuropixels 2.0. The two main things relevant for this talk are that it's smaller. You can see that it's just a physically smaller device, and also that it has 4 shanks rather than a single shank, and that enables recording more densely from individual brain regions, so more neurons per brain region, essentially. I won't describe those things. Here's what the Neuropixels 2.0 device looks like. You can see this again is 1 centimeter long, so you can see that the whole device is just a few centimeters long, and 2 probes plus the head stage that they connect to. This all weighs less than, or right about 1 gram. And so this is actually small enough to implant on the head of a mouse, and they can carry it around easily, and you can record in a freely moving context. Here's just another sort of graphic about what these Neuropixels 2.0 devices look like. This will be the production version on sale next year. The data quality is still high. I guess I'll skip over this a little bit. This is just some more example data traces now from the 2.0 probe. And this is the sort of payoff of the 2.0 device. This data from Chetay Aydin and Sebastian Hessler's lab, one of our collaborator labs on this project. They inserted 2 of these probes in a mouse brain and recorded and implanted them chronically. So they sort of insert the probes, but then cement them in place so that you can record while the mouse is actually freely behaving, walking around in its cage. And this spike raster you see here now is the abstracted version of those raw data traces. Now each little tick mark you see represents one of those action potentials that we saw before. Let me zoom way back here. So each one of these little events is going to get a single dot on this new plot. And so we're sort of zooming way out relative to those raw data traces. But now we can see, even in just a 2-second clip of data, we can see the hundreds and hundreds of spikes across probably hundreds of neurons on these probes. And this in total, I'm not going to go into sort of the details. There's some sort of details about how this recording was done, but essentially this is recordings from 6,000 recording sites in a freely moving mouse. And just to emphasize this point about the density of recording in an individual area, here we're seeing how having the 4 shanks helps you sample more densely in a brain region that has a size that's on the spatial scale of these 4 shanks. So this brain region is the striatum. It's the input nucleus of the basal ganglia in the mammalian brain. And these orange strips show where the recording sites were that recorded these 150 neurons. And again, this spike raster shows the timing of all those individual action potentials from each of those 150 neurons. And the point here in this data from Gentle Park and Josh Dudman is that this sort of apparent sequence of activity, this sort of horizontal or slanted stripe of pattern of activity that you see here repeatedly on every individual trial of the behavior, that really comes out when you have as many neurons as you can measure with this probe, and it's hard to see if you have fewer neurons sampled. So let me just summarize about Neuropixels. So this is a large-scale electrophysiology device using CMOS fabrication technology. That's what enables, like, using relatively modern fabrication technology is what enables the device to be so much higher channel count and to have that amplification and digitization system built in. 2.0 devices are miniaturized. They have 5,000 sites on 4 shanks and optimized for stable long-term recordings. I didn't talk about this, but I'm putting it here so that any aficionados in the audience can check out the paper and read about it. We tried to address with this device and with the new algorithms that we introduced in the 2021 paper, a solution for one of the main data quality problems with electrophysiology, which is when the brain moves relative to the probe, and we think we can correct for that problem. Any questions about Neuropixels before I turn to the science now?
I have a quick one. So when I was a little confused, you actually had a neuron label, like a number. But when you stick these probes in a region, aren't the probes picking up activity from an unknown set of neurons? Or am I missing? Are they actually able to single out individual neurons since you numbered them? It's like some aggregate over some unknown set of neurons, no?
Yeah, that's right. So let me go back again to the raw data here. Great question. So if, when we run our algorithm to process these data and we get, okay, this was one spike time, this is another, and all of these events look the same, therefore we think all 3 of these came from the same neuron, then this putative neuron that generated these spikes gets an arbitrary number. And what we know about that neuron is a couple of things. First of all, we know where it was recorded on the probe. We know it was on these channels as opposed to these channels, which are deeper in the brain. So we know something about its spatial location in the brain. And also know just a little bit about the biophysics of the neuron. You can see that, for instance, this neuron has this very broad waveform, and this neuron has this very sharp waveform, much more brief in duration. And that tells us a little bit about the ion channels that each of these neurons express. So we have limited information, and certainly the neurons in the mammalian brain are not intrinsically numbered. It's not like a C. elegans or even a Drosophila where you can identify the same neuron from one subject to the next. So we're instead getting a random selection of neurons, but when we run our preprocessing, we can sort of assign an arbitrary number to each of them. And so in this figure that you're referring to with this neurons numbered from 1 to 154, they were all recorded at sort of random positions around this probe, but have been reordered and renumbered according to the timing of their activity pattern. And that's what it means by sorted order here. Does that answer the question?
Yeah. I was just wondering, to what extent can you, if this is some sort of deconvolution problem or some sort of reconstruction, like to what extent can you associate a given signal to a given given neuron? It doesn't have to be obviously a number or anything, but just whether you're, is a given sensor sensing some number of neurons rather than, and then you have some post-processing allows you to disentangle this. So probably a really long answer to it, but I just, are you able to do single neuron resolution? Yeah, that's ultimately.
Yeah, we believe we are.
Okay.
And the problem that the algorithm is solving is essentially the one that you can sort of solve visually yourself here, right? Which is that we assume that a given neuron has a unique spatiotemporal pattern of activity that will be detected across the recording sites when it fires an action potential. And the reason we assume that is both because neurons do have these different biophysical characteristics, but also because the exact spatial position of a given neuron relative to the probe is going to determine the amplitude of its signal on the different channels that are recording it, because those each have their own position. So it's like the sort of cocktail party problem where you have a room full of microphones. The microphones are our electrode sites, and each neuron is a voice. And so we can tell by which sites have the higher amplitude detection of each signal, roughly where that neuron was, and that's the sort of information that we're using to figure out which spike goes with which neuron. So, like this event here is clearly a different one than this one and this one, whereas these two both look really the same, and they also look the same as that one and that one. And so that's what the algorithm is looking for, and it's finding matching events that it then characterizes as all coming from the same neuron. Does that make more sense?
Great, thank you. Yes, please. That's perfect. Thank you.
Yeah. All right. All right. So any other questions about Neuropixels before I move on?
And I just wanted to mention that, as Nick said, but just like to emphasize that this is really kind of like created a revolution in neuroscience in terms of ability to record not just from a single neuron or several neurons, but actually to record from like whole population of neurons down to single neuron scale, and also over time, over behavior and so on. So this set of tools is, and hardware and the analysis that comes with it to identify the neurons and kind of like to filter the data are really revolutionizing neuroscience and behavior research.
Yeah, thanks, Ellie. Yeah, and by the way, the problem that I was just describing to Mark about how to process the data, that's called spike sorting. It's kind of a whole field to itself, and we have an algorithm that I participated a little bit in making. I was mostly the work of Marius Pachtaru, called Kilosort, and there are a couple of papers about it. You could really go into depth on that problem. It is an intense data processing problem, but I'll skip over it mostly for now. Cool. Okay, so let me move on to the science. So to introduce the kind of sort of neuroscientific questions that I want to ask, I've got this little video, and I think we can see a couple of interesting things about mammalian behavior in this video. So you see that there are two deer, and one of them at some point in this video, it detects some sensory signal and orients towards it. He looks over towards presumably the photographer here. The other deer walks on completely unaware, and so it seems that they will have likely experienced almost the exact same sensory stimulus, and yet the one deer detects it and the other deer fails to detect it. So one question we can ask, a simple question is, what led to the difference between this deer detecting and orienting towards this stimulus versus this deer not detecting, or at least not orienting towards the stimulus? Is it something, for instance, about the brain state of this deer? Maybe he's on high alert because it's his social responsibility at this moment in time to be the one looking out for predators or something. And the second thing is this moment where the deer sort of stops chewing, and we can almost anthropomorphize that there's some interesting process going on inside its head. And actually, it's a very critical one, a totally life or death process, where this deer is trying to take into account all of the sensory information available to it, weigh it all up, everything it can see and hear and smell, weigh up all that information and determine whether this is a threat, in which case he needs to sound the alarm and also get the heck out of there. Or instead, is this something harmless, such as some geeky National Geographic photographer, in which case you can ignore it, which is the decision the deer apparently makes here. And so we can ask, what exactly is going on inside the brain of the deer when they make this sort of species survival critical decision of weighing up all the different sensory information and deciding about whether to respond to it as a threat versus not. And so to summarize those two questions, you could ask, why are some stimuli perceived and others are not, even if the stimulus is apparently identical? And we call that the detection problem. Or we could ask about discrimination. How are multiple pieces of sensory evidence combined to make a choice and make a decision? And in the context of this talk, the particular aspects of these key and really basic perceptual and decision-making behaviors that I want to go after is to ask this question: are the correlates of these processes distributed or localized? We can certainly, so this is getting back to this complex wiring diagram that I put on the first slide. We can certainly imagine that it might be the case that a detection process simply has a single area collecting sensory information of a certain modality and either triggers a detection and an orienting response or doesn't, and you don't need a whole brain to do it. You just need one particular area which has that one particular role, and as long as that one area is doing its thing correctly, then you'll orient towards the stimulus, or not. And that's the question we're going to ask. So just to point to some pre-existing literature, which I think is interesting and sort of suggestive of what the answer might come out to be, this classic work from Ranulfo Romo's group recorded over many subjects and many recording sessions, recorded dozens of neurons from each of these different brain regions you see labeled here. This goes from primary somatosensory cortex called S1, which is a sensory receptive area, to primary motor cortex M1, which is an area that projects down to the spinal cord, and includes also all of these other areas, which are things like medial, pre-central cortex, prefrontal cortex, et cetera. These sort of quote unquote higher areas. And in this series of studies, this group, I think, hypothesized that they would indeed find that there's sensory processing at one end of this chain, motor processing at the other end of this chain, and one of these areas in the middle, the task was really to find out which one of these areas in the middle represents the decision process and actually carries out that decision to act on the piece of sensory information and produce a desired motor output. But that's not at all what they found. Here, I'm not going to describe this plot in too much detail. Suffice it to say that each row is a different brain region, same as the labeled regions over here. Each column is a different time point during a sensory discrimination task, and the different colors are the different types of neuron coding properties that they found in these regions. So again, I'm not going to describe the X and Y axes of these plots, but suffice to say that you see all of the different colors, so the red, green, and blue color in all of the different areas, except for this very top area of S1, where uniquely these signals are not so distributed. So it seemed to be the case that these correlates of this perceptual decision-making process, when authors looked across cortex, multiple cortical areas, were indeed distributed across those areas. And what we think we can do to go beyond this is to consider not just cortex, but also subcortical areas, essentially try to consider the entire mammalian brain and really ask, to go beyond this particular finding, ask whether the authors had simply not been looking at the right areas versus is this the story across the whole mammalian brain? And so to do this, I'm going to talk about this work that was collaboration with Peter Zakahas, a really talented graduate student, and Matteo Carandini and Kenneth Harris's lab. This is a project that I did as a postdoc. And in this project, we have mice. They are surrounded by 3 computer screens that we can show visual stimuli on. The mice have a wheel that they can turn clockwise or counterclockwise with their forelimbs. You'll maybe remember this from a video that I showed earlier, and I'll show you that video again. And mice are rewarded with water rewards when they get the correct answer. That is when they turn the wheel in the appropriate direction according to which stimuli they see on the screen. And the mice are head fixed while they do this. They're stuck in place, and that both, of course, allows us to combine this task with the neural recordings with Neuropixels, but also allows us to precisely control the visual stimuli that the mice are seeing. And the first version of this task was published in 2017 by Burgess et al. And so what do the mice see on the screen, and what are they supposed to do with the wheel? They can see a stimulus on the left screen, in which case they should turn the wheel clockwise to get a reward, or they can see a stimulus on the right screen, in which case they should turn the wheel counterclockwise to get a reward. And when they turn the wheel, the stimulus is actually going to slide across the screen according to which direction they turn the wheel. So another way to say the task is to bring the stimulus to the central screen, which requires the opposite wheel turn depending on which side the stimulus started on. Now, on some trials, there can be 2 stimuli, one on left and right, and then they have to choose the higher contrast stimulus. And this makes it a visual contrast discrimination task. They're trying to weigh up the 2 pieces of sensory evidence that they have in front of them to make a decision about whether clockwise or counterclockwise is the correct option to earn a reward. And in this way, you can see that this is a sort of weird and highly abstracted version of the same kind of discrimination task that the deer was doing in the video before. And then finally, there's a third type of trial where no stimulus is shown. And if that's the case, if the mouse doesn't detect any stimulus on the 2 left and right screens, then they can still earn a reward, but only if they hold the wheel still for 1.5 seconds for a short delay period. And this comparison between these no stimulus trials and the left and right trials, this is what makes it also a detection task. So we're querying the mouse about whether they detected a stimulus, in which case they should turn the wheel at least one of the two directions, versus didn't detect any stimulus. And we can ask this, use this to ask about the neural correlates of that detection process. Okay, so the mice learned to perform this task very well. So in this plot, the right side of this axis is when stimuli are on the right at higher and higher contrast, which is basically the detectability or stimulus strength. And on the left side of the plot is when stimuli are on the left. And right in the middle of this dot is when there's no stimulus on the screen. And the orange dots indicate the proportion of trials in which the mouse chooses to select the right stimulus, which is that counterclockwise wheel turn, given the different contrast conditions. And you can see that they're doing that correctly when there's a stimulus on the right side. And likewise, they typically correctly choose the left stimulus when there's a stimulus on the left side, but not when there's no stimulus on the left side. And finally, they succeed in making this no-go response, holding the wheel still for that 1.5 second delay when there's no stimulus shown, but do it rarely when there are stimuli shown, indicating they're successfully reporting whether they detected or did not detect stimuli. And you can see that they have some errors. For instance, these 30% of trials, when there was a low contrast stimulus on the left, 30% of the time, the mouse failed to detect that stimulus, even though it was an identical stimulus to the stimulus that was presented on the other trials. Okay, and this is a summary of all possible combinations of contrast. I'm not going to go through this in too much detail, but in this plot, we were looking at just one stimulus on the left or one stimulus on the right. But you remember I told you that there could be two stimuli at once, and so this grid is all the different possible stimuli on the right, all the different possible stimuli on the left, and showing that they trade off their left and right choices according to the difference in contrast between the left and right side, and they make this no-go response when there's no stimulus. Okay, so you saw this video before, but now you have some idea of what's happening on the left side here, where the mouse is doing this behavioral task. That was a no stimulus trial, and this is a stimulus trial with two stimuli. The mouse correctly chooses the higher contrast stimulus, spins the wheel to bring it into the center of the screen, and gets a reward. And so now we can ask what's happening in the brain across all of these neurons across many brain regions while the mouse performs this visual contrast detection and discrimination task. And are those detection and discrimination processes localized versus distributed? So to summarize the data set, we made recordings from 10 mice, about 40 recording sessions. And in total, this enabled us to sample about 30,000 neurons. And each one of these colorful lines is showing the 3D position of one of those recording spans where we recorded those 400 recording sites along a 4mm span through different parts of the mouse brain. And the impression you should get is that even over a relatively small number of subjects, we were able to sample a pretty good fraction of the total volume of at least one hemisphere of the mouse's brain. And in particular, we sampled these brain regions. So most of these brain region acronyms and names may not mean much to you, but let me say what a few of them are. So the blue ones back here are visual cortex, primary visual cortex in the middle, and secondary visual cortical areas. Up here we have frontal cortex and primary motor cortex, as well as somatosensory or touch cortex. Then if we look at a sagittal slice, so this is slicing the brain like this, if you can see my video, and look at some of these deeper structures, we have in purple parts of the basal ganglia, we have in green parts of thalamus, in pink parts of hippocampus, and in red parts of the midbrain. And don't worry, obviously, if you don't have a wealth of background knowledge about exactly what each of these brain regions has historically been associated with, because don't worry, I'm going to tell you what they do in this task. So to start asking what the neurons do in this task, let's take a look at an example neuron. So this neuron was recorded in the deep layers of primary visual cortex. This was the waveform that was detected, the signature of this neuron spikes by the probe. And this is a summary of the activity of this neuron. So here's a raster plot again, where again, each dot represents a single spike, a single action potential emitted by this single neuron. And each row now is not a different neuron like it was in previous plots in the first section of the talk, but each row here is a different trial of the task just looking at activity from this one single neuron. And so you can see this neuron likes to fire lots of spikes. When you have a high contrast stimulus, it fires a bit fewer when you have a medium contrast and less when you have a low contrast stimulus. And when you don't show a stimulus, this neuron hardly fires at all. Its firing rate appears to be at about the baseline level. And so we can summarize that rate by sort of averaging across trials, and that gives us these continuous traces shown down here, where we can see that the neuron is indeed firing in a contrast-dependent manner, so higher responses and also earlier responses for the high contrast stimuli relative to the low contrast stimuli, and responses only when there is a stimulus present versus not. So this is unsurprising because we already had many papers in the literature telling us that primary visual cortex is a place where you see visual responses and responses in visual tasks. So that's great. So let's zoom out to look at what's happening in the rest of the brain when we run this task. So in order to zoom out, I'm going to take this average activity over time of a given neuron and turn it into a row in this plot. So each row in this plot corresponds to a single neuron, and the color of the row going from white to black to gray indicates the activity of the neuron going from low to high to some medium level. And so we can see that there are many neurons in primary visual cortex up here that have this property of responding robustly relative to the baseline activity after visual stimulus onset, which is happening at the dashed line at time 0 during this task. So that's primary visual cortex. Here are some secondary visual cortical areas. And again, these areas had all been reported to have activity in response to visual stimuli and in visual tasks. And so again, this is not a surprise that they're responsive during this task. But if we zoom out and look across the brain, we saw something that surprised us. So if we look at frontal cortical areas, if we look at hippocampus, if we look at basal ganglia, if we look at thalamus, and if we look at midbrain, we find neurons in all of these regions that are responding robustly following stimulus onset in this task. So this is, and at a glance, there's very similar activity patterns in all of these regions. And in the subsequent slides, I'm going to break down what exactly are these activity patterns related to in the task. But at a glance, we can certainly say that this is consistent with this idea that the connectivity is so distributed and interwoven across all of these different areas that signals are highly distributed rather than being localized in a single area. So let's try to break it down and see if that turns out to be true. So in order to break down the activity patterns of each individual neuron and understand what aspects of the task they respond to, we are going to take advantage of the fact that we have different trial types in this task. So we have trials in which stimuli are shown on one side of the screen versus the other. This neuron is recorded in the left visual cortex, so contra here means shown on the right side of the screen versus the left side of the screen. We have trials in which the mouse successfully detected and responded to those stimuli, trials in which the mouse failed to detect and respond to the stimulus. And then we even have trials in which the task is over, the mouse is done, there's no opportunity for rewards anymore, but we show the stimuli again in this passive context and just see what the mouse's brain does in response to the stimulus, even when the mouse is completely checked out and not paying attention to or reacting to the stimuli at all. And we can see that for this neuron, at least in this early period of its response, we get a robust response when contralateral stimuli are shown, irrespective of the behavioral condition, irrespective of whether the mouse successfully detects the stimulus, fails to detect the stimulus, or whether the mouse isn't even engaged with the task whatsoever. So this neuron we would characterize as having a visual encoding. Its responses, at least in its early part, appear to be entirely driven by the visual stimulus itself and not by any other aspects of the task like the behavioral performance or the detections. And this property of visual encoding in a kind of positive control sort of sense, we found in visual cortex, both primary and secondary visual cortical areas. We found in frontal cortex, which receives direct projections from visual cortex. We found in the striatum, which again receives direct projections from visual cortex. We found in these two visual thalamic nuclei. And then interestingly, relatively less known, we found in a few midbrain nuclei that had in some cases been reported to have visual responses and in other cases not. But we didn't, for instance, see any visual responses in the hippocampus, which was, or somatosensory cortex, which is consistent with what's known from the literature about visual responses. But how about the behavioral responses? So here's a neuron that has a different pattern of response. If you show stimuli in this passive context, or if the mouse fails to make any detection of the stimulus, this neuron is silent relative to baseline. It just continues on its activity, baseline activity unperturbed. But if the mouse successfully detects and responds to a stimulus, irrespective of which stimulus it actually was, whether it was a left or a right stimulus, this neuron becomes active. And these black dots here are the times at which the neuron starts responding to the stimulus. So this neuron we would categorize as having what I'll call an action correlate. That is, it's correlated with the onset of the action or the action initiation. And this action encoding, we very surprisingly found in essentially every brain region we recorded from. So you have neurons in all parts of cortex, essentially, as well as all parts of hippocampus, thalamus, midbrain, and basal ganglia, responding in this way, this sort of encoding of the motor action that the mouse undertook. But what about a specific response to the motor action? What about the choice? What about the decision process? Deciding to turn the wheel clockwise or counterclockwise, that's the whole game. That the mouse is trying to play and win to get rewards. So are there neurons that have that property? Yes, there are. So here's an example of such a neuron. This neuron was recorded in midbrain area called the SC, the superior colliculus. And when the mouse chooses the contralateral stimulus versus chooses the ipsilateral stimulus, now this is aligned to the movement onset, the time at which the mouse starts turning the wheel, not aligned to the visual stimulus onset. When the mouse starts turning the wheel counterclockwise versus clockwise, this neuron has a very different response. So it's selective for the particular action or selective for the choice of the subject. It responds for counterclockwise but not clockwise choices. And here's another neuron in frontal cortex that has the same property. You can see it's responding on the opposite. It's responding on the ipsilateral ones, the clockwise turns of the wheel. Here's a neuron in the basal ganglia that has this property. Here's a neuron in another part of the midbrain reticular nucleus. And here's a neuron in an area called the zona incerta, an area that has not been previously studied in this type of behavioral task, but which nevertheless we discovered has neurons with really, really clear and large responses that are choice selective or action selection selective in this task. So here is, in the familiar breakdown of trial types that we were looking at for the other two example neurons, here's an example of that zona incerta neuron showing that it really responds right around the time of movement onset and in a choice specific way, only for one choice and not the other. But interestingly, we noticed that this property of having this choice selective response, though we did find neurons that had this property in lots of different brain regions, at least in a fair number of different brain regions, this response property was not the same everywhere. So in particular, we noticed that in midbrain areas, here at the top, midbrain reticular nucleus and superior colliculus, in midbrain areas, we nearly always saw that the neurons preferred or responded more on the orange trials, which is these contralateral choice trials. Whereas in forebrain neurons here in frontal cortex and in basal ganglia, this is secondary motor cortex and caudoputamen or the striatum, these neurons instead responded about 50-50, preferring orange versus blue, that is preferring clockwise versus counterclockwise choices. And moreover, it was common to see in the midbrain neurons that they were suppressed below their baseline for their non-preferred choice. You can see the blue dipping below the baseline level for many of these neurons, whereas in the forebrain neurons rarely did that. They were usually increased above baseline for both choices, but just more for one choice than the other. And so we called this a unilateral encoding scheme in the midbrain and a bilateral encoding scheme in the forebrain areas like frontal cortex, basal ganglia, and parts of thalamus. And so this was interesting because this choice signal is distributed. It's not the case that there's just a single area that represents the choice, but it's not distributed everywhere. And moreover, it's not distributed equally in the sense that there are different characteristics of that choice signal in different areas. So we asked this question, are the correlates of detection and discrimination distributed or localized? And I think you can see that the answer is distributed. Only the action initiation signal, the green map from before, was really, let me zoom back to it, was really distributed everywhere. But the vision and choice signals were distributed across multiple brain regions as well. So choice and action are represented broadly across the brain. But these are both sort of motor correlates. They're both about the actual action that the mouse is undertaking. And we can imagine that there might be specific reasons that motor correlates are distributed across the brain. Maybe, the mice are needing to alert the rest of the brain about the action that's being undertaken, so that all sorts of systems across the brain can adjust to the new behavior that the mouse is performing. So what about something like, what about something else? Is this in general true that anything we look at is distributed broadly across the brain? What about abstract representations? So here we're going to ask about a different kind of representation and ask whether it is also distributed broadly across the brain. And this project, which is so far preprint, but otherwise unpublished, is the work of two postdocs in my lab, David Ottenheimer and Anna Bowen, in collaboration with Garrett Stuber's group and a graduate student of his, Madeline Hort. And in this project, we're looking at the abstract concept of value. So what do we mean by value, and in what sense is it abstract? We mean a very specific thing, which is whether the mouse thinks that a particular sensory stimulus is likely to lead to reward or not. So in this case, we have odor presentations, not visual stimuli this time. We have odor presentations, which can either be a particular type of odor that always leads to reward, a particular type of odor that never leads to reward, or a particular type of odor that leads 50-50 to reward versus nothing. And in this case, we would say that the mouse, if the mouse succeeds in learning this task, the mouse will have a high value attributed to this stimulus and a low value attributed to this stimulus. And we can see that the mice do indeed learn this task and do indeed learn those valuations, because when we present the odor here from time 0 to 1.5 seconds, the mice actually start licking, even though the reward associated with the odor delivery is not presented until 2.5 seconds. So they don't get the reward until out here at the green line, but nevertheless, they start this process of anticipatory licking, which is an indication that the mouse expects the stimulus to be rewarding. And you can see that indeed the mice don't do that anticipatory licking on the CS- trials, the ones that always lead to no reward. So this is great. The mice appear to learn the values of these different stimuli and appear to act on them. But in what sense is this value actually an abstract value? I said I wanted to study an abstract representation. This could just be, an odor-specific reward signal, or maybe it just likes this odor, not maybe some neuron just likes this odor, not that odor. Well, we can look for an abstract value signal by training the same set of reward contingencies for a second odor set. So we have odor set 1, and now we have odor set 2, 3 completely different odors, but that have the same reward associations. And now we're going to look for any neurons that have the same responses on odor set 1 and 2 as indicative of them actually representing the abstract concept of value, divorced from the particular odors that carry those values. So in this experiment, we used Neuropixels again to record across areas of frontal cortex and olfactory cortex and motor cortex, as well as some areas of basal ganglia. This is the recording distribution of the study. We again see here in this plot that's very similar to the one I showed you before, where each row is a neuron and they're separated by their different brain regions, that we get activity in this task across many different brain regions. So I'm going slightly fast because I'm running out of time, I believe. We have a data processing, analysis method that allows us to separate out the neurons that are responsive to the stimulus per se, versus ones that are responsive to the licking itself. So I showed you that the anticipatory licking behavior is associated with the value, and we wanted to study neurons that have representations which again are abstract relative to concrete factors like the licking behavior. So we have a sort of GLM style model in which we can identify which neurons are responsive to the licks versus the cue presentation itself by using this sort of held out test in which we fit a model that tries to predict the activity of the neuron and ask whether the model is impaired if you don't tell it information about cues, licks, or rewards. This is a pretty simple, it's a linear model, it's a pretty simple model, but allows us to tell which neurons have cue related responses. Let me skip that plot. And we indeed find cue-related responses. So these are putative sort of odor or value coding cells in blue here across multiple different regions, including parts of motor, prefrontal, and olfactory cortex. Similarly, we find these lick encoding neurons across all of the regions as well in red. And this is replicating this finding from the previous study about distributed coding of action initiation. So this slide has the final result that I'll show, and then I'll summarize. We looked then for putative cells that encoded the value, and these cells, if they're going to be value encoding cells, they're ones that don't respond to the licking per se. We cannot predict the activity of the neuron with the licking, so they were excluded on the previous slide, but they have activity that scales according to the value of the stimulus. So higher activity relative to baseline, in this case, it's higher negative activity of this particular neuron, it's a downward deflection. For CS plus trials, the high value stimuli, and lower activity for CS minus trials, the low value stimuli. And it should have the same responses on set 1 as set 2, and that would indicate this sort of abstraction of the value signal relative to the particular odors that gave rise to it. We have this shuffle analysis, which I guess I won't describe in detail, but it attempts to control for how often you're likely to see a pattern like this by chance. And the result is that we see about 33% of those Q responsive neurons having this pattern of having this abstract value coding. And these other bars represent the other iterations of this shuffle, which are different types of encoding that are not associated with the value of the stimulus per se. And so these 33% of the neurons are the ones that we will then characterize as having abstract value representation. And here is their distribution across brain regions, where we can see that, in fact, the height of this blue bar, or the number of value encoding neurons that we find, is actually surprisingly uniform across all of the regions that we sampled, including motor cortex, prefrontal cortex, and olfactory cortex. And so we found that even if you're looking at an abstract conceptual variable, in this case, the value or expected reward of a stimulus, those signals too appear to be distributed broadly across brain regions. So just to conclude, I showed that Neuropixels CMOS electrode arrays or probes have enabled unprecedented scale across the entire mouse brain, at least across the entire mouse brain. They've also been used in other species without loss of resolution. And I gave you a couple of principles that organize or describe the behavioral coding across the brain. Motor processes, including action initiation and action selection, or what I call action and choice, are encoded broadly across the brain, though choice is encoded differently in midbrain and forebrain regions. And even abstract behavioral variables such as value is encoded approximately equivalently across diverse cortical and basal ganglia regions. And so taken together, these findings, I think, indicate that when we look at that convoluted spaghetti wiring diagram that I started the talk with, and ask what kinds of mental models and what kinds of experimental approaches should we have when approaching a system like that, it certainly could have been the case that we got lucky and everything was routed into very specific and modularized organizations, and that in order to study something like a visual discrimination choice, or to study something like abstract value coding, that we could just ignore most of the brain and focus on one particular brain region. But I think that these results suggest that that's not the right approach. We are unlucky. We do not get to narrow down our focus to a single brain region at a time. And instead, the mammalian brain has correlates of behavior broadly distributed across many brain regions, which I think prompts a different experimental and conceptual approach to understanding. So thanks to the funding sources, and happy to take any questions.
I had a quick question.
Yeah, Mark, and then Tom, I see, has his hand up too. So go ahead, Mark. Yeah.
Oh, I'm sorry. I'll wait for the hand.
That's okay.
Yeah, and I just wanted to chime in and thank Nick for joining us and giving such a great overview. And yeah, with respect to time, we are kind of like a little bit short, so we will go to several, like just a few of questions. And Tom has his hand up, so let's start with Tom's question.
Sure. Do you see variability from mouse to mouse, say that they've learned this task, but maybe they learn it differently, so that when you make a measurement once, you may not get a full sample and you have to try it again and again and see if it reproduces?
Yeah, great question. So in this study, we looked at the curves of performance that I showed you for an example mouse, and they were roughly the same across mice at least a first pass. And so we just made the assumption for the particular study that I showed you that all of the mice had learned approximately the same thing. But I think your question is a great one, and it's certainly worthy of further study. And the International Brain Lab, which I mentioned briefly before, has just had a preprint earlier this year asking exactly that question. So in that study, we had multiple mice recorded in each of 10 different laboratories, all trained on the same behavioral task and all recorded from the same spot in the brain. And in the paper, we sort of analyze what aspects of the neural data are reproducible across those different mice and different trainings. And I don't think we've quite done all the analysis you could do to ask, about the influence of behavioral strategy on those correlates. But suffice it to say, a lot of the neural correlates that we record with these probes are highly reproducible, despite any differences in behavioral strategy that might exist.
I guess if it's very spread out, then if it's differently spread out, it still looks spread out. So,
yeah, true. So we'd absolutely love to be able to sample sort of all regions from all mice, and we're not quite at that point with these probes. So maybe 3.0.
okay.
Yeah.
Okay. Thank you. Great.
Yeah.
Great
Thanks,
talk.
Tom. Mark.
Yeah, I was
And then
wondering,
Eric.
speaking of probe 3.0 or maybe some later version, are these probes, or is there any R&D around, I mean, as much as they pick up currents, can they drive currents? I mean, it might really mess with the mice brain, but like you could imagine, maybe also in the spirit of the Institute, you could, if you knew, you sense some behavior or some neural activity, you could imagine disrupting certain activities to try to, maybe there'd be additional understanding. So can these things, are they only probes, or can they eventually like drive currents as well as pick up currents?
Yeah, so you're hitting the nail on the head with respect to how neuroscientists like to approach these problems. What we would call manipulations or perturbations of the activity are absolutely a key tool in the toolbox. I didn't get to talk about another paper from my postdoc with the same authors, Zach Haas and Karen Dini and Harris, in which we did do manipulations of activity in all of the different cortical regions that I showed you, and perturbing the activity or shutting off the activity in each of those regions. We could ask which ones of them were critical for the mice to be able to perform the task, the visual discrimination and detection task that I showed you. So I can point you to that paper to get one answer. As for whether the probes can stimulate, certainly not these current versions. Other versions of probes are under development, and there are other technologies to manipulate activity, and that's absolutely a key direction.
Yeah, it seems like simultaneous, tightly integrated, pickup and driving might give you an even newer tool to be able to study. I don't know, but integrated in the same kind of device, maybe.
Yeah, and then the specific challenge there is that while you can absolutely do electrical microstimulation in mammalian brains, and you can change the activity of neurons, it's very difficult. I don't think anyone has like a real serious system, not that I know of, to target individual neurons or individual patterns of neurons with electrical microstimulation. And so even if you could drive current through the same recording sites that we're recording from, there's not really an expectation that you would be able to change the activity of the specific neurons within the population that you might be interested in manipulating. Instead, people use optical techniques for this, so you can optically read out the activity of neurons and optically manipulate their activity through genetic engineering tricks, and that has this property of being single cell resolution of both the readout and the manipulations. It has other challenges, like you don't have access to deep structures in the brain, and you don't have millisecond temporal resolution. So they're kind of complementary approaches.
Thanks. And then maybe since I have a hand up, another quick one is that to what extent is the density of these probes limited by the readout? I mean, I saw you had, kind of, I don't know, it fixed the readout chip, which is pretty sizable compared to the probe. I mean, are there exploration of, or would you benefit from readout technologies like RF or something that minimizes the footprint of the actual device so you could have a higher density?
Yeah,
are you not limited by that?
The limit on the current one is literally still the feature size at which you're printing the tiny wires that carry the analog signals from the sites up the shank and out to the recording chip. So that fundamentally limits how many channels you can get on a single probe currently. And so to try to improve that, literally, like, a project that is ongoing NIH funded project is trying to just use a smaller feature size fabrication technology, which is more expensive and comes with more noise challenges, but ultimately is like pretty straightforward to just scale down, and therefore scale down the feature sizes, and therefore scale up the number of recording channels available.
Okay, thank you. I'll put my hand up. Thanks a lot.
Eric, one more, or
Another hand is raised by Eric. Let's quickly
last one.
get to your
Yeah,
question
go ahead,
as
Eric.
well.
I'll be quick.
Hi, thanks for the fascinating presentation and results of your research. The one thing that I was wondering is whether there is any information in the pulse shape, and whether basically what you've shown is mostly, let's say, the impulse response of neuropixels.
Yeah, there is some information in the shape of the electrical signal detected from each neuron. I think I mentioned briefly that this is related, like the particular shape that each neuron emits, or that you detect from each neuron, is related to the biophysics of that neuron. Each neuron has a different complement of ion channels that determine exactly what its action potential shape is and what it looks like to an extracellular recording device. So there's just a little bit of sort of cell type specific information available to the probe. We can use that to categorize the neurons broadly by their biophysical properties, but it's not too much information.
Do you use it in what you've shown as a result of this research?
I did not use it in what I showed. Yeah.
Thanks.
Yep.
But yeah, it might be kind of an aspect of future research, right? Of kind of sophisticated analytical approaches that will be able also to encode and decode signal based
absolutely.
on
We have
various features.
Exactly. Yeah, we have an ongoing project where we have a version of the probe where it has the same number of recording channels, but much smaller and denser sites. So you can detect in much more detail the spatiotemporal pattern of the extracellular action potential. And that's exactly the kind of thing we're trying to do there, is see if, given that additional detail, if we can say more about the cell type of each recorded neuron. And no, we do not already have a catalog of the waveform shapes. Yeah. We're working on that. That's what this other project I mentioned is really about. So I agree. An important question. Thanks.
Yeah, this is super exciting. We're unfortunately running out of time. But yeah, I would like to, on behalf of all participants and members of the Institute, I would like to really extend a huge thanks to Nick for joining us for the colloquium. And we already see kind of like, I suspect that participants already see like relevance to the methods that they're developing and so on. So I'm hoping this will be a start of ongoing discussion with the UNIC, your group, and in general with the neuroscientists that are recording and analyzing these kind of like large scale recordings in terms of the future challenges, in terms of like real-time methods that will be able to respond as the recording is going, and also on even larger scale, as you mentioned, Neuropixel 3.0. So analyzing a huge amount of data.
Yeah,
These
exactly.
are the methods that are being developed, and in other disciplines are showing successes.
Perfect.
Yes, so several participants had to leave, but this discussion is being recorded, and I would like to encourage everyone to reach out if you have additional aspects to discuss to us, neuroscience section in A3D3, and also to Nick directly, and yeah, hoping to have like future developments in these directions. Thanks so much.
Thanks, Ellie. See ya.